So, I'm going to be talking to you about the GI tract and some of its inborn mechanisms or inbuilt mechanisms for keeping the instrument going and for protecting it because it is challenged. It's a cruel world out there. So to go back historically, in the beginning, there was not a void, but Auerbach, Meissner, Bayliss, and Stalling. So Auerbach did his work at the time of our Civil War. The North was fighting the South, and he was in Germany looking at the gut. And he discovered in the gut a huge, he described it, plexus of nerve cells. Meissner added a second plexus in 1888. And so at the turn of the century, Bayliss and Stalling, when they did their work, knew that when they were playing with the gut of a dog in a very drafty laboratory in London, and I know that those laboratories were cold because in winter, the indoor and outdoor temperature in the UK is usually the same. And one puts on gloves with fingertips open to keep going. And in any case, they were playing with the intestine of a dog. And what they found is that when they cut all the nerves to that dog's intestine and manipulated it by increasing pressure inside or pinching it, the gut always responded with an oral contraction, anal relaxation, that was propulsive. And all the nerves were cut, so they attributed that activity to those millions of nerve cells that they knew existed in the bowel, or what they called the intrinsic nervous system of the gut. So here's one of their tracings from that time. And here's the dog intestine. And at number 1, they pinch the dog's intestine. There's a balloon so they can see the pressure. And when they pinch this above the site, then distally there's a relaxation. When they pinch above, there's a contraction. So they see that there is an ascending process of relaxation and contraction of the bowel, and they call this the law of the intestine. So their conclusions, and here's their original work. Two kinds of movements are to be distinguished in the bowel: rhythmic pendulum motion and true peristaltic contraction. The pendulum motion, they said, was just the muscle. It did it on its own, and that turned out to be right. And Auerbach's plexus, the local nervous mechanism, that was propulsive, independent of the central nervous system. And that oral contraction, distal relaxation, is the law of the intestine. Well, unfortunately, a little bit later, Trendelenburg, a German scientist, during World War I, as trenches divided the continent of Europe and great armies were paralyzed for 4 years, killing each other, Trendelenburg had TB. And because he had TB, that kept him out of the trenches, even in the German army. But he had to make a living and keep busy. So he strung up a loop of guinea pig intestine in a test tube in vitro. And he blew into the intestine, and the intestine blew back. And this was amazing to him, because this little piece of guinea pig intestine demonstrated exactly in a test tube, when there was nothing there but intestine, the law of the intestine. But Trendelenburg called it the peristaltic reflex. I thought law of the intestine was kind of nice, actually. But anyway, we call it now the peristaltic reflex. So a little while later, John Newport Langley defined the autonomic nervous system. That's Langley. He looks severe in this picture, and he was known to everyone at the time as a son of a bitch. That was because he was the editor-in-chief of the Journal of Physiology, which he owned, which was then and is still one of the leading physiology journals of the world. He left it to the society, the British Physiological Society. But as editor-in-chief, he was an imperious editor. And people often found editorial changes made in their manuscripts when they appeared in print. This did not endear Langley to the crowd. So we generally think of the autonomic nervous system and attribute it to Langley. That's the autonomic nervous system, that's Langley, and this is the classical view of it. Sympathetic, parasympathetic. Sympathetic is thoracic or lumbar in its outflow, thorax, lumbar. Parasympathetic, cranial, sacral, and chain ganglia in the organs. So that's the view I was taught when I went to medical school, and it's classical. Classical means medical for wrong. In fact, that wasn't what Langley said at all. What he said, actually, is that the autonomic nervous system is an entirely motor division, that there are peripheral ganglia, and it has 3, not 2 divisions. The third division is the enteric nervous system. The sympathetic, thoracolumbar, parasympathetic, craniosacral. But enteric neurons, he understood, those millions of neurons that Auerbach had found, have no connection to the central nervous system directly. Only a tiny fraction of the neurons of the gut have any direct connection with the CNS. That's unlike any other organ. They're on their own. And so it's capable of integrative activity. And he understood that around the turn of the previous century, that is 19th to 20th, or the early part of the 20th. So this is the way we now understand the nervous system. And I'll go through very quickly for you. So there's the nervous system. It has a central and a peripheral division. The central nervous system is the brain and spinal cord. The peripheral nervous system, everything outside the brain and spinal cord, has a motor and sensory division. Motor carries information away from the CNS and gives rise to motion, glands secreting, and so on. Sensory carries information back. If I step on your toe, you know about it. Sensory. You notice that the autonomic motor system and the skeletal motor system are 2 parts. Skeletal motor system operates skeletal muscle. And that skeletal muscle, the nerves go directly from the CNS. They don't pass go, no synapses. So when it's determined in the CNS that your biceps is going to do this, your biceps does it. No peripheral modification. In the autonomic nervous system, there are peripheral ganglia. And those are sites where regulation occurs. So it isn't determined when the signal leaves the CNS that's going to happen. Your heart may not speed up. Your blood pressure may be modified along the way. Now, you notice there are 3 divisions here, and this one is colored yellow and blue, or blue-green. That's because it's got intrinsic sensory neurons within it, so the gut can detect what's going on. When Bayliss and Starling pinched the gut, or blew into it by Trendelenburg, when the brain and spinal cord are in the garbage with the rest of the animal, nothing's there but bowel. And so it's able to sense it and know what to do with the information all on its own. So peristaltic and secretory reflexes are mediated by the enteric nervous system, and the enteric nervous system and the CNS communicate with one another. It's not just one or the other. And 95 or 90% rather of the fibers in the vagus nerve, the main pathway between the gut and the CNS, are sensory. So the gut is sending information back to the CNS. So this is the nervous system of the gut. It's found inside the bowel. There's the myenteric plexus, submucosal plexus, and it ramifies up into the crypts, innervating the lining of the gut, which is the mucosa. In these fibers, red here represents nerve cell bodies, green are glia. So the brain and the enteric nervous system project to one another reciprocally. And as shown on this slide, not only does the brain send information to the gut, but the gut sends information back. And so the stomach has fibers that project directly back to the spinal cord. And the small intestine projects outwardly to the gallbladder and pancreas, innervates the sympathetic ganglia. So if the gut doesn't like messages that the brain is sending through the autonomic nervous system, through the sympathetic nervous system, it can cancel them. And the colon talks directly back to the lower sacral spinal cord. Now, I apologize to you for showing this after lunch. You're probably wondering what this is. What you're looking at here is a colon. This is a colon from a woman with Chagas disease. Chagas disease affects 3 to 4 million people in our hemisphere. It's caused by the bite of the kissing bug, which bites at the inner canthus of the eye and puts Trypanosoma cruzi in. It causes a carditis if you survive that and get an autoimmune response, which can take out the enteric nervous system. And so what this is a pseudo-obstruction. It's called a pseudo-obstruction because there's no anatomical obstruction. It's just the gut doesn't move. And so if you cut any part of the enteric nervous system, the gut becomes functionally obstructed as if you tied a suture around it. So this is a slide that will give you an idea of what we're talking about. This is guinea pig distal colon, and we're putting an artificial fecal pellet in, and you notice that the fecal pellet has been detected by the gut. Even though there's no spinal cord or brain, the gut is propelling it along, oral contraction, anal relaxation, puff. It made it. And now if we take the intrinsic circuits of the gut out by adding tetrodotoxin, which blocks voltage-gated sodium channels, just stops the nerves from working, you put the pellet in, nothing. So that's the second brain. It requires integration to make it work. A part that was relaxed has to now contract to move that down the gut. Now, one of the major chemicals that is absolutely critical to the functioning of the bowel, and the brain too for that matter, is serotonin. Serotonin is best known as a neurotransmitter. If you don't have enough of it, you commit suicide. If you don't have enough of it, you tend to be depressed. Antidepressants make it work better, and so we think of serotonin as happy medicine, a happy molecule, a molecule that smiles. And so years ago, when I sent an application to NIH, it went to a study section concerned with cognitive neurophysiology. And I thought, oh God, they're taking the second brain literally. However, when I got the comment back, I got one that said, this seems like an interesting set of experiments. However, and when you see however in a study section report, you know you're dead. It said, however, we do not understand why Professor Gershon chose to study serotonin in the gut. Well, the reason, in part, is that for serotonin, the gut is the big enchilada, to paraphrase Richard Nixon, who called that his attorney general, used the term. So quantitatively, the brain has got about 2 to 3% of the body's serotonin. Platelets have about 2%. 95% is in the gut. So my thought was, could the fact that 95% of the body's serotonin be in this organ be a clue to the investigators as to why it might be interesting? Like, did God put it there for sport? Turned out God didn't, not sport. So most of that is in the EC cells, which are the enterochromaffin cells, which are epithelial cells in the lumen of the gut. Small proportion is in enteric nerve cells. But if you add up the entire bowel, there's more total serotonin even in the nervous system of the gut than in the entire brain. Now, to be sure, serotonin in the brain is what makes life worthwhile. I mean, it's involved in sex, it's involved in temperature regulation, involved in sleeping, dreaming. I can't tell you what serotonin isn't involved in. That's easier. But in the gut, it's been a little more difficult. But what it's key is to making the gut work. So within the gut, there are two major serotonin compartments. One, as I've said already, is the enterochromaffin cells, shown here with silver immunofluorescence, with what are known as footpads extending out. The granules that contain all the serotonin in electron micrographs are at the base of the cell, because when the serotonin comes out, it comes out the bottom and stimulates nerves that are close to it, but not within it. So it's a paracrine method of secretion. In the nervous system, there are nerve cells, and there are two types of tryptophan hydroxylase, which is the biosynthetic enzyme that makes serotonin, TPH1 and TPH2. TPH1 is in the EC cells, TPH2 is in the nerve cells. So this means that you can play with the genetics and take out one or the other and know what the two types of serotonin are doing. And nerve cell serotonin is much less quantitatively, but very important. So what is the mucosal serotonin doing? Well, the first thing it was shown to do is that it's released by pressure from EC cells to initiate the peristaltic reflex. When the guinea pig gut squeezed, saw the artificial pellet. By the way, it was brown. It's a plastic pellet. It's brown just for the sake of verisimilitude. When it sensed it, it was serotonin that did the sensing. So in a little model, here's a model of an EC cell. And here's an intestine capable of motion. And when you apply pressure to the EC cell, it lays a serotonin bomb, and the intestine takes off. So that's the peristaltic reflex. Complex. Now, this slide has a lot of data on it, but I'll summarize it for you quickly. There are two types of tryptophan hydroxylase, TPH1 and 2. And if you selectively knock out TPH2, you alter GI motility. If you take out TPH1, you don't. Well, I just told you it initiates the peristaltic reflex. What this tells you is that if serotonin's not going to do it, something else will. But what it means is that you put more pressure on, and then the nerves respond directly to pressure. I won't show you this slide, but what happens is that if you don't have TPH1 or serotonin in the EC cells, the gut gets very long, very dilated in time, and it hurts. But enough pressure, the gut will go, even with no serotonin in the EC cells. If it were my gut, I prefer to have serotonin in my EC cells than to have it expand unmercifully and have every motion painful. But there's a backup. But in the nervous system, you have no backup. So if you knock out TPH2, total GI transit is affected. Gastric emptying is affected. Colonic transit, small bowel and colonic transit are all affected. So you need to have it. Well, why is it so important for the nervous system? In part, it's a neurotransmitter, but also because it's important in neurogenesis. Now, to understand how you get disorders in the system, I have to say more about serotonin. So it's made from tryptophan. You get 5-hydroxytryptophan. This is the tryptophan hydroxylase step. Serotonin is then produced, and it's pumped by a pump into vesicles. That's called the vesicular monoamine transporter 2, VMAT2. It then comes out by exocytosis stimulating it. It acts on receptors. And then another pump called SERT, the serotonin reuptake transporter, pumps the serotonin back in. And it can either be reused or go to a mitochondrion to be eliminated as 5-hydroxyandoleacetic acid. But the point I'm making here is that SERT is the turnoff. Serotonin isn't broken down like acetylcholine to make it stop working. It's pumped away. Nerve cells put it out, and they take it back. It's a kind of action. Okay. So that means because the actions are SERT dependent, changing the activity of SERT changes the activity of serotonin. If SERT goes up, gets more active, serotonin gets to be less active. If SERT is less active, serotonin is more active. All of the antidepressants have this as a mechanism. Prozac does that. Now, a word about autism. Autism spectrum disorders are frequently involved with the GI tract. They're fourfold more frequent in individuals with, or GI disorder, more in individuals with ASD, and often forms of autism associated with aggression and self-injury. Serotonin is very important in that phenomenon. About 30% of individuals on the autistic spectrum have too much serotonin in the blood. That means their GI tract is abnormal because 100% of the serotonin in the blood comes from the gut. So we utilized a model in which an abnormal human variant of a SERT gene has been expressed in mice, and when that's done, miraculously, the mice look like they have autism, and their GI tract stops working. So that means you can reproduce autism and its relationship to the GI tract by putting a gene associated with human autism into a mouse. That's called reverse genetics. And the reason you want to do that is not to be cruel to mice, but you want to do that to learn how these diseases come about. So I'll tell you a little bit about this problem. This is the tails of a molecular yenta, a molecule that is into everything. That's, of course, serotonin. And this is a mouse that expresses this abnormal form of SERT, super SERT. Now, it's not good to be such a mouse. So, one more point. The nerve cells that come to the gut are, like most of us in the United States, immigrants. They migrate to the bowel from the neural crest, and they grind it to the gut and colonize the bowel. So this is the sort of the new world as far as these nerve cells are concerned, and they mature in what we can call the lower east colon. So nerve cells, neural crest-derived cells, colonize the bowel. And the very first one to develop happens to be serotonin. In terms of evolution, it's very early as well. And it innervates these still dividing other types of cell. And it influences not just their activity, but it influences their fate. Serotonin gives them information about how they will develop. So a subset of the neurons in the gut are serotonin dependent. Their development depends on serotonin. Now, what happens if there is no serotonin? Well, you can do that by knocking out tryptophan hydroxylase 2. Gut has all of the serotonin in the mucosa, just lacking it in the neurons. And when you do that, you have too few total neurons. And if you knock out here TPH1, here's the wild type. Sorry, here's my mistake. Here's dopamine type neurons, wild type, TPH2 knockout. Massive, here, total neurons. And if you look at the double knockout, it's the same thing as just TPH2. So neuronal serotonin is critical to the formation of the nervous system itself. And so when you look at these nervous systems, and I won't go through all these pictures, just make you think of Miro, looking at all these pretty colors. But let me point out, what it shows is that the later born neurons depend on serotonin, and they are specifically deficient in these animals. And these are some of the types of neurons that are deficient and are serotonin dependent. And you can find, okay, it depends on serotonin. There are some 20 some odd serotonin receptors. One class of serotonin receptor turns out to be the key, and it's the serotonin type 4. And that's interesting because there are drugs on the market. Prucalopride has just been introduced in the United States. It's been available in Europe and Canada for a while for treatment of IBS. And if you give that drug, what these slides show is that just adding a 5-HT4 agonist can mimic all the actions of serotonin. And if you block that action, you can take out the action of serotonin. Okay, keep that in mind. And now let's see what happens to the enteric nervous system if they knock out SERT. Well, if you knock out SERT, you tell me what's going to happen to the nervous system. If there's no SERT, then serotonin becomes more effective. If it becomes more effective, what will happen to neurogenesis? You have more or less? More. Overabundance. And that's what you get. Here's total neurons, dopamine, CGRP, GABA, They all go up. And so you just have a big, fat neuronal gut. Does it work well? No, it doesn't. Those neurons get all over one another. They can't work right. It's the Goldilocks phenomenon. Here is everywhere. Got to be just right, not too hot, not too cold, just right. So it's these late-born neurons. You can see this is total neurons. This is with the overabundance of SERT. Now, what happens if you look at a super SERT mouse? Now, let's see, super SERT. What happens in super SERT is that SERT is too good. It really sops up serotonin like crazy. So the nerves put it out, and SERT really goes before it can act. So it's the same thing as no serotonin. Serotonin is perfectly fine in terms of it's made. All of the receptors are there, but you're taking it away before it can act. And as a result, the total neurons and all these serotonin-dependent neurons are now deficient in that kind of animal. And how does the GI tract work? It doesn't work well at all. And so this is the autistic mouse. In terms of behavior, they put two mice, put them in a channel. Normally 2 mice, they have a negotiation if neither one can get by, who's going to do it. In this case, the SERT knockout, the SERT over super SERT mouse always backs out. 2 mice put in, they don't look at each other. And their gut is working very, very poorly. And so colonic motility is terrible. Total GI transit is terrible. And if you give serotonin, it is taken out. And if you look at the peristaltic reflexes by filming it, so that's the pellet movement. And so you can see the contraction waves of a normal intestine here. That's the red. The computer is fixing that as red. And the super SERT, there are fewer of them, and they don't spread very well, and so just not working. So now it turns out that the nerves also control the epithelium. And so the animals that have too much SERT, too little SERT, wind up with huge villi, because their nerves are potentiated, and serotonin and acetylcholine regulate the growth of the epithelium. Now you can fix the problem of SERT activity and normalize it. So here in a mouse, to review, the villi crypts are abnormal. Fluoxetine or SERT knockout, they get to be too big. Super SERT, they're too small. But you can take all of these problems of the super SERT mouse and fix them by giving prucalopride. So here's the submucosal plexus. You can fix it in topal neurons, dopamine neurons, CGRP neurons, my enteric plexus, GABA neurons, fix GI transit, fix the frequency, everything that's wrong, the isolated preparation, just by adding during gestation prucalopride. Simple drug over the store can cure it. And I'm not showing the data, but it also fixes the behavior of the mouse. Now, one of the ways the bowel fixes itself is to add new neurons. And so one way you can look at neurogenesis is to use a model in which we labeled the precursor cells, the neural crest-derived precursor cells, with a Cre, which is a molecule that cuts out a piece of DNA, and you prepare the mice to remove a stop signal in front of a reporter gene. So whenever you can activate Cre hooked to the estrogen receptor by giving tamoxifen. When you do that, the estrogen receptor goes into the nucleus, Cre cuts out the stop signal, and whatever gets activated becomes fluorescent. So when you do this and then cut out a piece of gut, you find that the precursor cells turn into neurons. And they do so at a much increased rate, and you can see neurogenesis. So here's a new neuron done that way, provoked by cutting the gut. This just shows that that new cell is a neuron. Here it is fitting in a ganglion. It's not a glial cell, which is marked here, and it fits right into the enteric nervous system. So you get neurogenesis in the adults. And this phenomenon is serotonin dependent. So serotonin keeps the gut functioning. It also determines the severity of intestinal inflammation. So if you potentiate serotonin by knocking out SERT, inflammation of the gut gets very severe. And you can protect mice from colitis or inflammation of the gut by knocking out serotonin from the EC cells. So the EC cells not only stimulate peristalsis, but they're hooked up to the immune system and they stimulate inflammation. And if you knock out SERT, you make that worse. In contrast, if you knock out TPH2, the nervous system of the gut, you do the reverse. You decrease the severity of inflammation. So mucosal serotonin is pro-inflammatory. Why should it be? Well, the lumen of the gut contains pathogens. The EC cells detect pathogens. They put out serotonin. That stimulates inflammation to fight infection. So the same apparatus that's used to trigger motility is also used for protection. And protection, when you get inflammation, that can kill neurons. So serotonin makes new ones, if that happens. So depletion of mucosal serotonin protects mice from colitis. And if you look at mice with super SERT mice, they're protected from inflammation because serotonin can't work. It's removed too quickly. Do those animals pay a price for that? Well, why did you think it evolved to have serotonin drive inflammation from EC cells? To protect the gut. So if it's not doing that, will it protect the gut? No. You just look at 16S RNA and you find in the mesenteric lymph nodes that there's more of it. These animals are constantly getting infected by the commensal organisms of the gut. And you can see it electron microscopically in these poor mice. Look at that, right there. God, look at that sucker caught in flagrante. Okay, on the other hand, if you deplete neuronal serotonin, you increase the severity of colitis, and you get massive releases of cytokines. So serotonin is the sword and shield of the gut. There are two sources of serotonin in the gut. The large mucosal store of serotonin in EC cells can be used to combat danger to the bowel. It's TPH1 dependent. It's present in EC cells, in mice, also mast cells, and it's pro-inflammatory. The small, little tiny enteric nervous system. That store of serotonin is equally important because it's active in motility, neuroprotection, neurogenesis, mitigating inflammation, protecting the ENS from it. And so it protects serotonin, the crown jewels of the gut, which are, after all, the enteric nervous system. So perturbations early in development, stress or infection, that alter the activity of serotonin neurons can exert lifelong effects on the ENS and GI function. And conceivably, the anecdote that kids with colic, infantile, grow up to be adults with IBS, or people who have very severe infections early in childhood can have long-lasting abnormality of the gut, comes about because it affects the early bowel. So I'm just one of a group, and I've had lots of collaborators over the years of that, particularly Cara Margolis, who's working with me now. Thank you.